Welcome to the Counterpoint Session C1 of the Fourth World Congress. This is the first major international meeting on alternatives to be held in this millennium, and it's quite fitting that at this time we stop for a moment not just to think about reduction, refinement and replacement, but to think about the need for animal research for scientific purposes at the present time. What I hope we can do today is not to go over the past, but to think about the present and where we might be going in the future. To do this today, we have two speakers who are familiar and well equipped by experience, qualification and training to provide informed insights and analysis and a range of views sufficient, I'm sure, to stimulate discussion. The format we'll follow here is that each speaker will have 15 minutes to set out their views of these issues. We will then, if we're on to time, of 20 minutes discussion led from the floor, which I will moderate, and at the end we'll give each speaker 5 minutes to sum up what they think has been said and what they think the key messages are. My role will be to keep to time, and unlike Jerry Springer, it will be to bring out the best in our speakers rather than the worst in them. And the intention is to generate light rather than heat. It's not to test the strength of people's views, the sincerity of those views, or the duration of time for which they've held those views. It's to actually hear and listen to the evidence and rationale for the views. Our two speakers today will be known to some or all of you. Ray Greek is a medical graduate who was an anesthesiologist and a specialist in pain management, and in addition to his clinical practice, he has experience of research both in man and animals. He is the president of the Americans for Medical Advancement, which advocates responsible spending on medical research of your tax dollars if you're an American resident. He's known best outside America, I think, from his publications, and certainly the publication of Sacred Cows and Golden Geese attracted a lot of attention in Europe and further afield. And his more recent publication, Species Science, is also now receiving attention and comment. The other speaker is Michael Festing. Michael is based at a UK Medical Research Council unit at the University of Leicester in the UK. And he's known to me as a geneticist, statistician and expert on experimental design. He is also a frame trustee and chairman of the Frame Reduction Committee. And in addition, Michael and I touch base from time to time, as Michael is also a member of our Animal Procedures Committee, the independent source of advice to government ministers on animals and science. And with those relatively few words, I'll now ask Ray Greek to begin his presentation.
Okay, thank you very much. Can you hear me in the back? Can you hear me in the front? Do any of you even want to hear me in the front or the back? That's the question. Well, thank you. Thank you all very much for coming today. Thank you, Dr. Michael Festing and the organizers of the Fourth World Congress for scheduling this session. And again, thank you and the audience for attending. Hopefully, this session will shed some light on our respective positions. Some time ago, I was asked by Dr. Festing to explain my reasons for believing that animals are no longer valid for modeling humans. During this debate, I will try to outline those reasons, but as it will be impossible to address all areas in 15 minutes, I refer you to the handout, which is available on the back table, to AFMA's website and to our books for a more detailed examination of this question, and to the handout specifically for a response to Dr. Festing's critique of our first book. It is easy to forget how far civilization has come in the past 300 years. Before Newton, demigods ruled the universe, and if crops failed or sickness afflicted one's family, it was assumed to be caused by disfavor with God. Darwin's theory of evolution, electron microscopes, DNA, the study of genetics, chaos, and complexity were unheard of. And I think I can say without fear of contradiction, at least from this audience, that the philosophy of life, known as science, has been responsible for these changes and the advances that so separate us from our ancestors of just a few hundred years. When society first started using science and animals in a serious attempt to learn about human disease and health, it made sense in a way. Grossly, animals and humans had things in common, important things. Both were composed of cells. Both were affected by viruses and bacteria, responded to vaccines, had circulatory systems, lungs, livers, and so forth. So the initial idea of extrapolating results from animals to humans appeared valid, and indeed society did learn things from animals. We can debate whether many of the great discoveries of old were dependent upon animals, or whether the animal model even then did more harm than good, but that is not why we are here today. Today we want to have a science-based discussion about the animal model in 2002. If one grants that initially animal models were useful, then the next logical question becomes, what has changed? Since this debate is science-based, it may be appropriate to take a moment and remind ourselves what separates science from non-science. In our books, the website, the handout, and, as you will see in my portion of this session, I will present evidence based on science, evidence usually derived from peer-reviewed literature. Science does not allow proponents to assume their conclusion, but mandates they prove it. I will quote data from the scientific literature and stay away from what could only be called opinion, as is required by science. In science, opinion counts for little, while references that can be checked are the currency of the day. Of course, science also concerns itself with predictability and falsifiability. So the first question to be addressed is this: is there a problem with animal models? Let's take ADMET as an example. First, some facts. Most drugs are effective only in 30 to 60% of patients. Adverse drug reactions are the fourth leading cause of death in the U.S. and the U.K., and 15% of hospital admissions are caused by adverse medication reactions. Legal drugs kill approximately 100,000 people per year in the United States, more than all illegal drugs combined, and cost the U.S. taxpayer over $136 billion in health care expense. Clearly, the probability of any given medication doing more good than harm is not optimal, and this, at least in part, is due to the animal testing that occurs prior to clinical trials. Tom Patterson, the chief science officer at Intellos, likens the current practice of drug testing in humans during clinical trials to, quote, making airplanes, trying to fly them, and marketing the ones that don't crash. Not a good reflection of the efficacy of the animal testing that occurs prior to human clinical trials. Barry Selleck, formerly with Glaxo, said that for every drug that is withdrawn from the market, 10 remain available to people even though they have ADMET-associated problems, and for every drug that is on the market with ADMET-associated problems, there is an additional 10 to 50 that will fail before they reach the general public. All this despite extensive animal testing. 80% of new chemical entities, or NCEs, fail after phase 1 clinical trials, and hepatotoxicity is the most common reason for this failure. Vis-a-vis hepatotoxicity, Mark Levin, PhD and CEO of Millennium Pharmaceuticals, presented this slide at a drug discovery and technology conference in Boston 1 year ago. 28 potential medications were tested on rats, and 22 went to be tested on humans regardless of the rat results. Of the 11 that were shown to be hepatotoxic in rats, 6 actually tested liver safe in humans. Of the 17 that rats predicted to be safe, 6 were shown to damage the human liver. Levin concluded that this means that the rat data was basically as accurate as a coin toss, not exactly the predictability we think of when we think of science. Clearly, the ADMET testing process, as it pertains to animal testing, is inadequate, as should be expected from a study of evolutionary biology and complex adaptive living systems. Attempts to design drug tests that only equal the success rate of animal tests will be of no benefit to society. Pharmacogenomics and broader, longer clinical trials offer the next step in the evolution of drug development, and that is where we should be focusing our resources. The fact that the 3Rs community continues to sing the praises of a failed modality, while some in the pharmaceutical industry admit its inadequacies, is, in my opinion, significant. This slide further illustrates the problems of complex systems and hints of extrapolation. Among 10 medications withdrawn from the U.S. market between 1998 and 2001, 8 were withdrawn because of side effects that occurred primarily in women. A recent study in Science revealed that one strain of mice could have a gene removed without obvious adverse effects, while a similar strain would die without the gene. If men cannot predict the effects of a drug for women, and one strain of mice cannot predict what will happen to another if a gene is removed, perhaps we have reached the level of organization or complexity that defines one species from another and even defines one individual from another. Evolution, the study of complexity, nonlinearity, and molecular biology combine to predict that animal testing should not work, and most importantly, empirical data, such as I have presented today and in our books, supports this. Most practicing physicians will tell you that the animal data is meaningless to them because it has no predictive ability, and hence they do not even read the animal experimentation literature. Likewise, physicians have complained for decades that longer and broader human clinical trials should be mandated. In the old days, people opposed animal experimentation for many reasons that turned out to be unsound. In the 1800s, for example, many who opposed the animal model also rejected science in general and anything associated with it, like the germ theory of disease and vaccines. Today, the thing that many in the 1800s anti-vivisection movement rejected, science, provides us the best reason to reject transspecies extrapolation. Today, the level of our study has changed from the gross level to the genetic, and at this level, evolutionary biology predicts that the amount of data we can extrapolate from animals has diminished since the 1800s. Even though humans may share 100% of their structural genes with another organism, say mice, these two organisms can be as different as a man and a mouse. By studying mice, it is obvious that we can see only a piece of the puzzle, a mouse piece, not a human piece. And the rest of the pieces are usually ignored to the detriment of humans suffering from illness. Evolution has not built each species from scratch, but has added on, as Toshny and Gann write about in their book, Genes and Signals, to an already complex system. The use evolution has made of add-ons has resulted in living complex systems that behave like the complex adaptive systems that they are, and as such differ significantly from their predecessors. Add-ons produce systems that are non-linear. Small changes on the genetic level, such as add-ons, can lead to very large differences between species. Indeed, that is what evolution is all about. The claim that humans and mice are the same animal at the biochemical level, just dressed up differently, isn't true. Moreover, it is irrelevant to point to observed similarities in genetic makeup between species, since the details of the differences are in the interactions between the conserved genes, not in the genes themselves. It is as though humans and mice have a common genetic keyboard on which different phenotypic tunes are being played. What matters is not the similarity with respect to the keyboard, but the differences with respect to the order and timing of the pressing of the keys. Predicting human response based on an animal model is not an example of, quote, applying a relatively simple set of well-established scientific principles, end quote. Living organisms are better examples of complexity theory than Newtonian physics. Using the example of a model airplane or an automobile, as so many defenders of the animal model do, is a good example of this. Studying a model of a 747 will allow the observer to demonstrate the basics of flight, just as studying a 1973 Vega will demonstrate the basic principles of an internal combustion engine. But if anyone seriously believes that the model airplane or the Vega can be or is used to repair a Ferrari or a 747, they are deluded. Just as animal models can be and were used to demonstrate very basic facts concerning anatomy and physiology, so a Vega and a model of a 747 can be used. But today, when we want to fix a Ferrari or repair a 747, we don't tear down a Vega engine, nor do we use a model airplane. And neither should we suggest to the public, who is paying for animal experimentation, that a cure for AIDS, cancer, stroke, and so forth will be derived from animal models. A recent article in New Scientist said, quote, another key finding from both public and private genome efforts is that many human transcription factors are unique and a cut above those of the fly and the worm. Transcription factors and other regulatory proteins dictate which genes are switched on at vital stages of development as embryos form and organs take shape. It is they that orchestrate such amazing complexity from so few genes. Vintner, and the article is referring to Craig Vintner of the Human Genome Project, Vintner thinks all higher vertebrates have roughly the same genes. What is important is when they are switched off and on. He says, quote, we have the same number of genes as cats and dogs, but differently regulated. Researchers such as Marilyn Carroll and Bruce Overmeyer, in their book, Animal Research in Human Health, state that animals are causal analogical models and thus can be used to study human disease and predict human response. Note this is different from using animals merely as heuristic devices. Causal analogies are a subset of analogy arguments in which causal assumptions arise based on the model. La Follette and Shanks have explained how models are used as cams in their book, Brute Science. Basically, if a drug, Z, causes death in an animal model, animal experimenters will reason by analogy that it will also cause death in humans. Animals are thus used as causal analogical models, and this reasoning process has resulted in immeasurable human harm, as we and many others have pointed out. In light of our current knowledge of DNA, evolution, molecular biology, complex adaptive systems, and nonlinearity, I conclude that the burden of proof is now on those who claim animal models are predictive. They should produce prospective data gathered by investigators without a vested interest in the outcome that proves animal test A, B, and C, and so forth is reliable. It is not incumbent upon us to provide any more data supporting the theory that animals are not furry-looking humans. Darwin, Watson, Crick, La Follette, Shanks, Kaufman, Collins, Vintner, and many others have provided more than enough data. Using animals as cams, causal analogical models, in 2002 makes a complete mockery of the scientific method. In closing, I would ask Dr. Festing these 4 questions. Are animal models used as cams in drug testing and the study of human disease? And if so, why do they fail so miserably in ADMET? And if not, how are they used in drug testing, as they can certainly not be considered heuristic? If you claim they are heuristic, why does Big Pharma and other vested interest groups claim they are cams? Number 2, how specifically do we know which findings in which animals can be extrapolated to humans? For example, most species are unharmed by thalidomide, Regulin, fen-phen, oprin, practicalol, and so on. And why do interspecies variation, as predicted by evolution and complexity theory, allow for this extrapolation? Number 3, can you produce peer-reviewed data proving that animal models are predictive by comparing prospectively obtained animal modeled results from all species tested to the actual human results? And finally, number 4, if the purpose of the animal model in biomedical research is to aid in easing human suffering, how do you explain the fact that 2 leading causes of death in developed countries, a high-fat, high-cholesterol diet and cigarette smoking, were both deemed safe in animal models, that the animal model influenced French scientists to allow HIV-infected blood to be transfused, and that the animal model delayed the implementation of life-saving modalities such as artificial heart valves, cyclosporin, penicillin, Lasix, the statins, the SSRIs, and so on? In other words, what ratio of harm to good is acceptable in a model? Thank you for your attention, and I welcome any questions during the Q&A.